today I'm going to tell you about some very recent work from my lab, which we've been doing over the last year. And it's about analyzing protein language models and understanding their generative properties and their data representation. I'm going to start by telling you a bit about the context, and then I will move on to analyzing the generative properties of a specific protein language model, which is called MSA Transformer. And next, I will tell you about how this model represents phylogeny. Before moving on to all this, let me tell you a bit about protein sequence data. So we already had a talk about this, so I'm going to go a bit fast on this introduction. But to remind you, proteins can be described at different levels. The basic level of description is the sequence of amino acids in this heteropolymer, so which amino acids are present at each site. And in principle, if we know the sequence, we know everything about the chemistry of the protein, and the sequence fully determines the structure in 3D that the protein takes, as well as the function, which is its biological role in the cell. But on the other hand, the function and the structure have a big impact on which sequences can actually survive natural selection, because selection is done at the level of function. A protein is selected to be a good enzyme or a good molecular motor, etc. And so when we look at the sequences that have survived natural selection and that exist in natural organisms, we have access to a footprint of this selection. Okay, so another exciting aspect of this data is that it's growing extremely fast. Here you can see this plot, which is in log scale in Y. So this almost linear growth means that there is an exponential growth of the number of available sequences. And here on this slightly older plot, which is in linear scale on the y-axis this time, you can see that while there is the big growth of the total amount of data, we in fact know quite little about this data from experiments. So for instance, in red, you can see the amounts of proteins for which we know the structure from an experiment, typically X-ray crystallography or other techniques. So it means that there is a lot of data, but most of it is unexplored by experiments. And this is a great opportunity for statistical physics, information theory, machine learning, etc., all methods based on data to learn about proteins. Another point I want to make about protein sequence data is that it can be organized into protein families. A family of proteins is a set of proteins that have evolved from a given ancestral protein and that have a similar function and a similar structure. And these sequences have similarity in sequence, and they can be aligned together. For instance, you can imagine that this is the hemoglobin protein, and the first sequence is the one from the human, the second is the one from the chimpanzee. This is actually a simplified example, but I'm just saying this for, to give an idea. So we are going to focus on these protein families. And now, let me tell you a bit about how machine learning is impacting this field. So I'm sure you've all heard about AlphaFold, which has made a very impressive progress in protein structure prediction from a supervised point of view. So it's a model that uses natural language processing techniques, specifically attention and the transformer architecture. And that is trained in order to predict structure using the experimentally known structures and sequences as input. And you can see here the progress that it made in the contest of structure prediction that takes place every 2 years. So AlphaFold won the contest in the last 2 editions, and the model that I was telling you about is actually AlphaFold 2. So it reached a score which was deemed to be the score to be reached in order to claim that the protein folding problem was solved, which is quite impressive. Okay, but in this talk, I'm not going to tell you about the details of AlphaFold. I'm going to rather consider this kind of architectures and to see how non-supervised learning methods using these architectures can inform us about proteins. And I'm going specifically to focus on a model which was introduced in this paper by Rao and colleagues, which is called MSA Transformer. And this model is starting from multiple sequence alignments of proteins that are homologs. And it has actually adapted the transformer architecture to this kind of data. So it was built using the masked language modeling objective, which means that if you start from an alignment of protein sequences that are homologous, so each row is a sequence, each column is a site, you mask randomly a certain proportion of the entries, and then the model is trained in order to fill in the masked entries. Specifically, it's trained to minimize the pseudo-likelihood loss, which can be written this way. And I would like to mention also that other models have been built in order to work on single protein sequences. But here I'm focusing on the one that actually works on multiple sequence alignments. Okay, and the architecture of this model is adapting a transformer architecture to this 2D structure of protein multiple sequence alignments. And for this, it uses something that is called axial attention, which interleaves attention on columns and attentions on rows. And the attention on row is tied with the idea that in order to fill in a masked entry in a row, you will need to attend to some specific amino acids which are in the same row, but these are going to be usually the same in another homologous sequence. So the row attentions and the column attentions are interleaved, and then there are also feed-forward layers, and all these is stacked multiple times. Okay, so let's now look a bit at what this model can predict. And something which is quite interesting is that this model, which was not trained in order to predict structure, actually has captured information about the structure of proteins. So this is, again, a result that was shown in this original paper that introduced the model MSA Transformer. And they looked at the ability of the model to predict structural contacts, that is to say, pairs of amino acids that are close to one another in the 3D structure of the protein. And they compared the model, so which is in blue here, to two other classes of model. ESM1B is a representative of these single sequence language models that I was telling you before. And POTS models are models which are looking at the co-evolution of columns in the alignment. And you can see here that MSA Transformer, which incorporates both types of information, is actually outperforming these two models at the contact prediction task, which here you have the percentage of success on multiple protein families versus the depth of the MSA, that is to say the number of sequences. It's easier to make predictions if your alignment has more sequences, and MSA Transformer really outperforms the other models by a lot for shallow MSAs. Okay, so that's a very interesting point, that these models capture contacts. In fact, they capture them in their row attentions, the tied row attention that I was telling you about before. So here, in fact, if you want to fill in these amino acid sites, you will have to attend to specific positions, and these positions tend to be usually the ones that are in contact, because there is co-evolution between these sites. So that's the idea. And now I'm going to move on to our work. And so the part I'm going to present now is a joint work with Damiano Sgarbosa, PhD student, and Umberto Lupo, who is a postdoc in my group, and they are both in the room. And it's going to be about generative properties of this model, MSA Transformer. So how can we generate new proteins using this model? And the idea is that we actually exploited the masked language modeling objective that is used for the training of this model in order to generate sequences from the model. For this, we start from a natural multiple sequence alignment, and we mask some entries at random, and we employ the model in order to predict the missing amino acids, but then we iterate by putting back this MSA as entry, as input of the model, and repredicting some masked entries, etc. After some iterations, the properties of the multiple sequence alignments stabilize, and we then consider these sequences as our generated sequences, with the idea that they are going to be sequences that are generated from the same family of proteins as the one that we put as input, but they are new sequences. And so then the question is, can we characterize these sequences, and can we compare them to sequences generated by other models which are generative? And for this, we compare them to POTS models, which are models based using co-evolution ideas. So here I'm showing you what a POTS model is. So a POTS model is giving you the priority of observing a given protein sequence in the protein family that you're looking at. It's a family-specific model. And its form is a Boltzmann distribution, which has one body terms, which depend only on one site, and two body terms that depend on two sites or two columns of the multiple sequence alignments, and that inform about the couplings between the columns and the co-evolution between them. So usually these two body terms have been used in the field in order to predict the contacts. And this was the kind of unsupervised contact prediction that you saw here in green, in fact. Okay. So these models are known to be good generative models. Proteins have been synthesized experimentally from these models and shown to fold and to have a good function, which is why we take these models as a kind of reference. And so let me show you now a comparison between the natural sequences and the generated sequences by both the POTS model and MSA Transformer. So here you can see two different types of score. The first score is a hidden Markov model-based score, which is called the Hammer score, and it's a score that is used to determine homology in protein sequences. The second one is the score that is actually derived from the POTS model, which is the energy value, minus the energy value in the POTS model. In both cases, higher scores means that the protein is better, fitting better in the family. And we are plotting them here versus the Hamming distance to the closest natural neighbor to see if this has an impact. For instance, if our generative model is only producing sequences that are close and that have good scores, or if, on the contrary, we can find really novel sequences that have good scores. So you can see here in red the original data, cloud, and in blue the data generated by MSA Transformer. And you can see this interesting fact that MSA Transformer tends to generate sequences which have actually higher Hammer scores, which are thus predicted as being better homologs than some of the natural sequences. And also an interesting correlation which tells you that it's not because the sequences are closer that they are better, rather the more distant sequences tend to have better scores. So this is quite interesting. You can see also on the projected histograms that the overall scores tend to be better for the MSA Transformer generated method. And we can also compare this to the BM-DCA generated proteins, which are the ones generated by a POTS model. The sequences generated by the POTS model, on the contrary, have rather lower scores than the natural proteins. But we can also see something interesting, which is that they have more diversity. They are more different from the natural sequences. And in fact, the conclusions are quite similar when we look at this other score, which is the POTS model score, POTS model minus energy. We can see that MSA Transformer is producing proteins that have scores that are similar or better than the natural sequences, while the POTS model BM-DCA is producing sequences that have lower scores. Okay, so this was just for one protein family, which is the mitochondrial ADP-ATP carrier. Now I'm going to show you some results for more families. We have studied, in fact, 14 different deep protein families, which really correspond to completely different protein functions, etc. We are focusing on the deep ones, which means lots of sequences in the alignment, because we need this in order to fit properly a POTS model. Okay, so here you can see for each protein family, 3 different distributions in 3 colors, which represent the original sequences, the ones generated by MSA Transformer, and the one generated by the POTS model. And for each of these ensembles, we show 3 types of scores. So the first one is showing the Hammer score, the one I showed you for one protein family before. You can see that it's quite general that MSA Transformer generated sequences have better scores than original ones, while BM-DCA generated sequences have slightly lower scores. Statistical energies show the same kind of behavior, and we are also showing a score which comes from feeding the sequences that we generated to AlphaFolds and looking at the confidence it has into the structure prediction task. So this is called the PLDDT confidence score. And here the story is quite similar. Again, the MSA Transformer generated sequences have quite good scores. Another way to characterize these sequences is to look at the statistics of the alignments. Are we reproducing well these statistics, like the frequency of occurrence of an amino acid at a given sequence, or the frequency of co-occurrence of 2 amino acids at 2 different sites, etc. So let's first look at the second-order connected correlations, which are defined these ways. And here, okay, so we are showing the generated data versus original data. And if things were ideal, everything would fall on the diagonal. And here you can see the kind of dispersion that we would expect in the case, for instance, where original data and generated data would be just 2 halves of the real original data, which gives you an idea of the kind of minimal dispersion that we would get if everything was very good. You can see that the 2 models that we are comparing have slightly higher dispersion than this, but that they are falling quite well on the diagonal. The Pearson correlation coefficient is better for the POTS model than for MSA Transformer. but here the slope is slightly more close to 1 for MSA Transformer. If we look at our 14 deep PFAM protein families, results are quite similar. The Pearson correlation coefficients tend to be rather better for the POTS models, and the slope depends a bit on the family. So it's quite interesting. We see here that essentially the POTS model is slightly better at reproducing these two-body frequencies, and in fact that makes sense because the POTS model is trained in order to reproduce these statistics. Something that I haven't insisted on, but when these models are trained here, they are pairwise maximum entropy models which are trained in order to reproduce the one- and two-body frequencies, and these parameters HI and EIJs are fitted for this objective. So it kind of makes sense that the models that we are showing here, the POTS models, are very good at capturing these two-body frequencies. But now let's move on to three-body frequencies, which this time are not the training objective of the POTS models. So what we see for these three-body frequencies, when again we look at original data and generated data and plot one against the other in these 2D histograms, we can see that this time the Pearson correlation is much better in the case of MSA Transformer than in the case of the POTS model BMDCI. And the slope is also much closer to 1 for MSA Transformer than for the POTS model. And this means that this MSA Transformer is capturing some higher order statistical dependencies in the data that were not captured by the POTS model. We can also look at this in another way by looking at an information theory quantity that is essentially a generalization of mutual information to 3 random variables. It's called co-information. You can see here the distribution of co-information in original versus generated data. And again, the distribution is much better reproduced by MSA Transformer than by BMDCI, which concentrates around 0 co-information. Okay, so higher order statistics seem to be better reproduced by MSA Transformer. Another thing that we have looked at is the distribution of sequences in sequence space. So it's something that is quite hard to address. However, we have looked at this by doing a one-hot encoding of sequences and then looking at the 2 top principal components, which I have to say don't explain much of the variance, right? But nevertheless, we have looked at them. And you can see here the distribution of sequences in this space. And the MSA Transformer generated sequences have a very similar distribution to the ones of the original data, while BMDCI, which captures the overall structure of the distribution, is tending to smooth the distribution a bit too much. So again, we have this impression that MSA Transformer sequences are capturing better some higher order dependencies. So let me conclude on this part. We have proposed a method to generate sequences using MSA Transformer, which directly exploits the masked language modeling objective by iterating over it. And the generated sequences have good scores, which are, in fact, and perhaps surprisingly, even better than the natural ones on average. Then we observe that the one- and two-body statistics are better reproduced by the POTS model BMDCI than by MSA Transformer. But the opposite holds for the higher order statistics, such as the three-body ones. And to us, this all means that MSA Transformer is a good candidate to generate novel proteins from a given protein family. And it gives a perspective, which is related to one of our previous talks, in protein design. And okay, to finish on this, unfortunately, I don't have the preprint yet, but it should be coming extremely soon. All right, so now let me move on to the final part of my talk, which is still about this model MSA Transformer, but this time we analyze how it captures phylogenetic relationships. And this is joint work with, again, Umberto Lupo and Damiano Sgarbossa. Here, to motivate this part, let me tell you that, as I said in the introduction, MSA Transformer uses this axial attention, which computes row and column attention matrices, and the tied row attention matrices have been shown to capture the structural context in an unsupervised way. What about the column attentions? So in the original paper, it was shown that the column attentions, when you take some average of them, it correlates somewhat with the phylogenetic weights, which are 1 over the number of neighbors of a given sequence. And this was interpreted as meaning that more attention is given to more diverse sequences in the model. We asked a question which is going beyond this to ask whether the model encodes detailed phylogenetic relationships between sequences. And the idea of this is that it would be really attractive that the model has captured a kind of orthogonal representation where in the row attentions you have this information on the correlations from the contacts between the columns, and in the column attentions you will have the similarities from phylogeny. Okay, so let's look at these column attentions. And so here, I'm showing you all the matrices of column attentions. There are 12 layers and 12 heads in the model. So you have 12 by 12 matrices here. And each matrix has the same size, in fact, as a Hamming distance matrix for this particular multiple sequence alignment, which corresponds to this protein family. The Hamming distance matrix is giving you the distance between two sequences in each element of the matrix, just counting the number of amino acids which are different between the two sequences. You can see that there are some blocks in this Hamming distance matrix, and the blocks correspond to the fact that the sequences in this alignment are sorted according to phylogeny. So close sequences in phylogeny are close by, and they tend to have small Hamming distances. These are the blocks from the phylogenetic tree. So what you can see, and it's, I think, quite striking, that already by just looking at the attention heads, you can see similar blocks, right? It seems that these blocks from the Hamming distance are reproduced by some of the attention heads. And so we asked whether we can train a regression to predict Hamming distances from these column attentions. And we first asked whether we can do this within a given family. And so for this, we average the column attentions across columns, and we fit a logistic model to regress the Hamming distance matrix entries from the entries in the attention matrices. And we then test on a held-out portion of the Hamming distance matrix of this alignment. So you can see the results here for 4 different alignments, 4 different PFAM families. These are the regression coefficient on each layer and head of the model. And you can see it's already striking that some of them are firing consistently across the different alignments. And also that the early layers of the model are more important in these regressions. Okay, these are the norms here. And here you can also see the prediction. Okay, so here you can see a comparison of the ground truth Hamming distances, which are in the upper triangular part of these matrices in blue, and the predicted Hamming distances from the regression, which are in green in the lower triangular parts of the model. This is for the training subsets of the alignment, and this is for the test set, which was held out from the beginning. And you can see that we capture quite well these block structures from the phylogeny in these Hamming distance matrices. Okay, so these are just 4 examples. We have done this analysis for 14 different PFAM families. And here you can see the coefficients of determination are squared, which are very good for all these families. Then we wanted to ask, okay, you can see that some heads fire consistently. And in fact, it's something that we can quantify by looking at the Pearson correlation between the regression coefficients across different MSAs. And you can see here that these correlation coefficients are quite high. And given these results, we actually tried to train a global logistic model of the column attention matrices to predict the matrix of pairwise Hamming distances between sequences, but this time training one model on multiple MSAs at once and testing it on other MSAs. And you can see here figures which are quite similar to the ones before, except that this time it's the general model. And again, some heads fire, especially in the early layers of the network. And you can see again that the ground truth Hamming distances are overall quite well captured by the model. The coefficients of determination are nevertheless less good than before, and this is because we have a kind of deviation in the slope of predicted versus ground truth distances, especially for shallow MSAs. So it seems to be a bias for shallow MSAs. But overall, the model is still capturing very well these features of the Hamming distance matrices, which is quite interesting because it's a general model. It seems that MSA Transformer has learned to capture phylogeny in a quite general way, not specific to each MSA. So finally, the last part I want to address is whether, given that MSA Transformer is capturing these 2 types of information, contacts on one side and phylogeny on the other side, can it actually disentangle correlations from contacts and from phylogeny? And this question is actually a very important question in the field of coevolution of protein sequences, because when we observe natural data, we observe correlations between columns, but we don't know if these correlations are actually coming from a structural constraint, a functional constraint like this one, where you need to have complementary amino acids in 3D in order to preserve the structure, or if they are just coming from historical contingency, for instance, from a case where 2 mutations occurred quite early in the history, which would also lead to correlations between columns. So that's quite a fundamental problem in this kind of analysis. And in fact, in the case of POTS model, it has been discussed quite a lot, and it's known to be a problem for contact prediction. Many corrections have been introduced. And here we ask whether contact prediction by MSA Transformer is more or less robust than contact prediction by POTS model. Because it's very hard to disentangle correlations from contacts and correlations from phylogeny in natural data, we decided to generate controlled synthetic data, which has only correlations from couplings, and also synthetic data, which has correlations from couplings, but also phylogenetic ones, because we generate it on a tree. So here, on this left-hand side, we are showing data which was generated by Metropolis Hastings Monte Carlo sampling at equilibrium, and all sequences are independent, and here they are generated along a tree. We are showing here the reproduction of the contact maps. The upper triangular parts are the ground truth contact maps that we want to predict, and the lower triangular parts are the predictions. So here you can see that predictions are quite good, but when we add the phylogenetic noise to the data, you can see that there are much more false positives in red that appear, and that they are scattered across all the contact map. This is true for this family, but also for that one, and in fact we have looked at 14 of them again. So that's what happens with the POTS model. These are predictions of contacts from the POTS model. You can see that phylogeny is bad for the prediction by the POTS model. Okay. Now let us compare this to what happens for the same synthetic data, but now when contacts are inferred by MSA Transformer using the row attention heads. And you can see here that the phylogenetic noise between these two cases, without phylogeny and with phylogeny, is really less important than here. We don't have all these scattered false positives, and it's the same for this family. So quantitatively, we can compare PPVs or AUCs, and we can see that MSA Transformer is more robust to phylogenetic noise than the POTS model. So to conclude on this, MSA Transformer captures phylogenetic relationship within column attentions, and so it has learned phylogeny and contacts in orthogonal representations in the model. We also find that thanks to this representation, MSA Transformer is more robust to phylogenetic noise than POTS models in the unsupervised contact prediction task. So an interesting perspective, in my opinion, is whether MSA-based language models could be used to infer phylogenies. And so if you'd like to know more, we have recent preprints on this topic. And finally, I would like to thank my co-workers on this project. So in particular here, Umberto Lupo and Damiano Sgarbosa, and also all my group members. And I also thank colleagues for discussions and discussion groups at DPFL, as well as my current funding sources. And okay, here is a picture of my group to finish. And I thank you all very much for your attention.
Thank you very much.
Thanks.
Are there questions? One here? Yeah, you can also just take mine.
Thank you. That was a fascinating talk. And my question refers to other modalities that you could use. So have you thought, or do you plan to use other information embedded in these sequences? So, for example, codon identity or chemical property of the amino acid. And do you have idea how would it affect your models, and how would it maybe it would improve it or provide some information where the, let's say, codon identity is even more important than amino acid identity?
Yeah, thanks for the question. I mean, I think it's a very exciting point. I mean, so here I should specify that the model is trained on amino acid sequences. So it doesn't know about the codon identity, but I think it would be super exciting to look at this in the framework of another model. I'm sure it can provide extra information, especially when we look at evolutionary points such as phylogeny. This could have much more information. So I think it's a very good, very interesting point. Regarding the chemistry of the amino acids, I think it's, again, a super exciting question, which kind of is related to the previous talk that we saw before the break. Yeah, I mean, I would say exciting future direction.
Okay, thank you.
Over there.
Thank you for the talk. Did you try to mix the two models, by the way? So in the last, using the POT sort of constraints?
Sorry, to mix the POTS model with the...
MSA Transformer. So using in the loss of the MAC the POTS conditions, but the free one, not the 2-1.
Sorry, the free one?
If you go backward to your slide, yeah, go ahead. When you compare the free distance.
Yeah, sure.
Yeah.
So this, like, for instance, this comparison, yeah?
Yeah, exactly.
Right.
Yes, this one, the free one, so EGK.
Yeah.
So I think it should be very interesting to use this as a loss, an additional loss constraint.
I see, okay, yeah. Okay, so within the POTS model, this is something that has been discussed, like whether, okay, so the POTS model here is trained in order to reproduce 1 and 2 body frequencies, and because of this, it has only 1 and 2 body terms in the Hamiltonian. Indeed, adding a third order term is possible. However, there is a technical difficulty here, which is that in order to learn properly this, you need really very long multiple sequence alignments, and you have a lot of parameters, so it's something that hasn't been so much explored, I think, because of this problem. Purely practical problem. But I agree that it would be super interesting because, I mean, there is this fundamental question whether there are 3 body terms which are, not only encoded in 2 body terms. Yeah. So thanks a lot for the question.
Other questions?
First of all, thanks for the talk. And then I had a question related to the fact that many times you actually use Hamming distances in your graphs,
You're right?
Have you tried to actually, for example, look at the similarity between amino acids, for example, blossom-based identity substitution matrices?
Yeah, that's a great question. I mean, it's true that here, for instance, for the phylogeny part, we are looking at Hamming distances as a kind of rough measure of phylogeny, like the simplest one that we can look at. I agree that blossom matrices would be very interesting to take into account chemical properties in more detail. It's something that I think Damiano has explored a bit. So yeah, we are looking at it, but for now we are mainly, I mean, we started by Hamming distances. Yeah.
Especially in a previous graph in which basically you show that more diverse generated sequences perform better, right? One possible yeah, it's like way back.
Yeah, it's before. Yeah, here, right?
That one, exactly. Maybe one possible explanation could be that the model on the left basically switches more amino acids, but like by not going far, right? It's like
yeah, that's a very good point. I think it would be very, yeah, we should look at it. Yeah. Thanks a lot.
Thanks. Other questions? Okay, so then thank you again.
Thank you very much.
Talk.